Hypersensitivity. Introduction. The immune response is generally a protective process, but it may sometimes be injurious to host. The hypersensitivity refers to a condition in which immune response results in excess reactions leading to tissue damage, disease, or even death in the sensitized host. The hypersensitivity occurs in individuals who have had a previous contact with antigen allergen. The initial contact sensitizes the immune system, and the subsequent contact with the same antigen causes hypersensitivity. Classification. The hypersensitivity reactions are classified into two main types, immediate and delayed types. The major difference between the two hypersensitivity reactions are shown in the table. The comb and gel classified the hypersensitivity reactions into four major types, type 1 to 4. Later on, type 5 hypersensitivity reactions was also described. Type 1 anaphylactic reactions. The anaphylaxis is the classical examples of type 1 hypersensitivity reactions. It is a IgE mediated reactions. Type 1 reactions occurs in two forms. The acute potential fatal systemic form called anaphylaxis, and the recurrent non-fatal localized form called atopy. The mechanism of anaphylaxis occurs when a sensitized individual come in contact with the shocking dose of antigen. An interval of 2 to 3 weeks is required between the sensitizing and shocking dose, during which the cytotropic antigen antibody IgE, previously known as a reagenic antibody, produced against the antigen attached to the surface receptors of mast cells and the basophils. The IgE molecules binds to these receptors by their Fc, and once sensitized, the individual remains so for a long period. When a shocking dose of the same or immunologically related antigen is given, the antigen combines with the cell bound IgE antibody on mast cell rapidly. The antigen antibody complexes stimulates mast cells and basophils to release the mediators that causes a clinical manifestations of anaphylaxis. The chemical mediators are of two types: primary mediators, which are the performed contents of mast cells and basophil granules, and the secondary mediators, which are newly formed open stimulations of mast cells, basophils, and other leukocytes. The primary mediator includes histamine, serotonin, eosinophil, chemotatic factor, and of anaphylaxis, proteases, and hydrolysis. The secondary mediator includes source reacting substances of anaphylaxis, prostaglandins, thromboxane, platelet activator factors, and cytokines. Atopy runs in families. These individuals have a tendency to produce a reagent antibody in unusual large amount. The reaction occurs at the site of entry of an antigen. Inhalation of pollen affects lungs, bronchial asthma, ingestion of a fish, milk, egg, drugs, gastrointestinal disorder, or cutaneous eruptions, and contact leads to local allergy, conjunctivitis. It is IgE mediated hypersensitivity reactions. IgE is homocytotropic, that is species specific. Only human IgE can fix to the surface of human cells. The atopen combines with the cell-bound IgE antibody, fixes on the surface of the mast cells and the basophil, and this antigen antibody stimulus complex stimulates these cells to release the mediator, resulting in clinical feature of atopy. Examples of atopy includes food allergen, dust allergen, and drug allergen. Type 2 reactions, cytotoxic reactions. The cytotoxic reactions is mediated by IgG or rarely IgM, the antibody direct against antigens on the surface of the cell, resulting in cell damage. The antibody binds to an antigen on the cell surface and causes a phagocytosis of the cell through opsonic or immune adherence. Cytotoxicity by natural killer cells, lysis through activations of complementary systems. Examples includes autoimmune anemia, hemolytic disease of newborns, and drug reactions. Type 3 immune complex reactions. It is characterized by deposition of antigen antibody complexes in tissue, activation of complement, and infiltration of polymorphonuclear leukocytes, leading to tissue damage. Two typical type 3 reactions includes Orthos reaction, localized due to reactive antibody excess and serum sickness generalized because of relative antigen excess. Authors reactions. Authors 1903 observed that with repeated subcutaneous injections of normal horse serum antigen into rabbit, the initial injections were without any local effects, but with later injections, intense local edema and hemorrhagic necrosis developed. This type of reactions is called orthos reactions. The orthos reactions can be passively transferred with the sera containing high titer of antibody IgG, IgM. Serum sickness, it is a systemic form of type 3 hypersensitivity reactions. It appears following a single injections of a high concentration of a foreign serum. The antibody to foreign serum reacts high enough titer by 7 to 12 days, but still some excess antigen remains in the circulating blood. The immune complexes get deposited on the endothelial lining of blood cells in various parts of the body, causing inflammatory infiltrations. Massive complement activations and fixation by antigen antibody complexes leads to fall in complement concentration. The serum sickness differs from other hypersensitive reactions in that a single injection serves both as a sensitizing and shocking dose. The immune complexes damage the tissue in the same way as that in orthos reactions. Some important immune complex disease or post streptococcal glomerulonephritis, endocarditis, and dengue hemorrhagic fever. Type 4 delayed or cell mediated reactions. The reaction is mediated by the sensitizity lymphocyte, which on contact with the specific antigen releases lymphokines that causes a biological effect on macrophages, leukocytes, and tissue cells. The type 4 hypersensitivity cause occurs within 48 to 72 hours of antigen challenge. As it is not antibody mediated, it cannot be passively transferred by serum, but it can be transferred by lymphocytes or transfer factors. Two types of delayed hypersensitivity reactions are well recognized: the tuberculin injection type and contact dermatitis type. Tuberculin infection type, when a small dose of tuberculin is injected intradermally in an individual sensitized to tuberculoprotein by prior injections or immunizations, and erythema and swelling. The induration occurs at the site of injections within 48 to 72 hours. The injection site is infiltrated by a large number of lymphocytes and about 10 to 12 percentage of macrophages. The unsensitized individuals, the tubercle injection provokes no response. The tubercle test is a useful indicator of for delayed hypersensitivity cell-mediated immunity to the bacillus. The positive tubercle test does not indicate present infections, but implies that the person has been infected or immunized by the microorganisms in the past. The cell-mediated hypersensitivity reactions developed in many infections with the bacteria, Mycobacterium tuberculosis, Mycobacterium leprae, fungi, and parasites. Contact dermatitis type. Delayed hypersensitivity may sometimes develop as a result of skin contact with the range of sensitizing material. The metal such as nickel and chromium, drugs such as penicillin or other antibiotics in ointment, such simple chemicals like hair dyes, picryl chloride, cosmetics, and soaps. These substances can act as a haptens after absorption through skin. These molecules combines with the skin proteins to become antigenic. The cell-mediated immunity is induced in the skin. The dermatitis is infiltrated predominantly by lymphocytes and few macrophages. Type 5 stimulatory type reactions. It is a modification of type 2 hypersensitive reactions. The antibody interact with the antigen on cell surface that leads to cell proliferation and differentiation instead of inhibition or killing. The antigen antibody reactions enhance the activity of affected cells. The typical examples in Graves' disease, in which the thyroid hormones are produced in excess quantity.